Welcome, everyone. Let's step straight into a world that is as fascinating as it is fatal. We are looking at the culinary minefield of natural toxins. From the land to the deep ocean, the everyday foods we rely on can harbor deadly, microscopic secrets. For the food technologists, clinical toxicologists, and forensic scientists joining us today, we are going to unpack the precise mechanisms of these toxins together. We'll explore how they compromise food safety, complicate clinical management, and leave behind distinct forensic signatures. Right, let's dive straight in.
So consider this rather unusual question. What do polar bears, pufferfish, and Haitian zombie powder all have in common? Well, the answer lies in the highly potent biochemical compounds they carry. You see, nature's deadliest toxins often hide right where we least expect them. In our everyday pursuit of nutrition, humanity has repeatedly stumbled upon biochemical defenses capable of completely shutting down the human nervous system. Let's examine the evidence, starting with a classic, rather chilling case from the ice.
Section 1. Toxic animal livers. Too much of a good thing.
We begin our journey on land. Early Arctic explorers discovered a terrifying reality about the polar bear liver. Unknowingly, consuming this highly nutritious organ poisoned them severely. This brings us to a fundamental principle of clinical toxicology. Sometimes the dose makes the poison. Just look at the staggering contrast in the numbers here. Ordinary butter contains roughly 2,400 to 4,000 international units of vitamin A per 100 grams. But the polar bear liver, it packs an incredible 1.8 million units. Consuming just a tiny portion delivers a massive, lethal dose. There's even a recorded case where a fisherman ingested around 30 million units from halibut liver. He suffered severe headaches, vomiting, and his skin literally began peeling off.
For the analytical chemists and biochemists with us, here's the exact pathway. Beta-carotene cleavage. Beta-carotene breaks down into two molecules of vitamin A in the intestinal mucosa. Now, we absolutely need vitamin A for normal growth and vision. It is essential. But extreme excess triggers acute hypervitaminosis A. The nutrient essentially transforms into a systemic toxin. It leads to cerebral edema, severe joint pain, skin desquamation, and ultimately death. It's a perfect illustration of how a vital biological molecule abruptly becomes a severe clinical emergency.
Section 2. Prions. Rogue infectious proteins. Let's shift our narrative from vitamin toxicity to one of the most terrifying biological mysteries uncovered in modern science. Back in the 1980s, global food safety sectors were absolutely paralyzed by transmissible spongiform encephalopathies. You probably know it as mad cow disease. But the culprit wasn't a bacterium, it wasn't a virus, it was identified as a prion. The term blends proteinaceous and infectious. For those of you in food processing and safety, prions are an absolute nightmare. Why? Because they completely resist standard sterilization protocols. They survive ultraviolet radiation. They even resist the body's natural proteases, those enzymes meant to break down proteins. Once they enter the central nervous system, they cause untreatable, universally fatal brain diseases.
To understand the exact mechanism, imagine a perfectly shaped molecular puzzle piece. The normal protein, PRPC, is structurally well-defined and completely harmless. It's found naturally throughout healthy bodies. But its infectious counterpart, PRPSC, is badly folded. It's polydisperse. This is the mutant form originally responsible for scrapie in sheep. And it's this precise structural misfolding that makes it so deadly.
So, the crucial point for clinical scientists is how this infection progresses. Let's walk through the steps. Prion interaction mechanism. Once the rogue PRPSC is introduced, it acts like a bad molecular influencer. It interacts directly with the normal PRPC proteins and catalyzes them to physically change their shape. They then aggregate extracellularly, forming rigid amyloid plaques. For veterinary scientists and pathologists, this structural disruption becomes visible under a microscope as literal spongy holes or vacuoles right there in the brain tissue. It's a relentless, fatal chain reaction.
Section 3. Pufferfish and zombie powders.
We now plunge into the ocean to explore marine neurotoxins. We start with the pufferfish, the second most poisonous animal in the world. This represents a highly unique cultural and culinary paradox. On one hand, you have the live takifugu pufferfish. Notice its rough, unassuming, sandpaper-like appearance. When threatened, it simply inflates with water. But on the other hand, we have fugu, the highly prized Japanese delicacy. The skin, ovaries, and liver of this fish are packed with lethal toxin, yet the muscle meat is elegantly served raw.
It requires chefs to be specially trained and licensed to ensure strict food safety. Just one wrong cut, and the dish is fatal.
And here is the microscopic culprit responsible: Tetrodotoxin, or TTX. Pharmacologists will immediately recognize this structure as one of the most toxic non-protein natural products known to science. It has a remarkably precise mechanism. Ingested TTX binds directly to specific sites on voltage-gated sodium channels in the nervous system. Think of it as a broken key that perfectly jams a lock shut. It completely blocks the influx of sodium ions, which instantly halts action potential generation and stops nerve transmission dead in its tracks.
The clinical timeline of TTX poisoning is, quite frankly, terrifyingly rapid. Within 5 to 20 minutes, the victim feels an initial numbness and tingling around the lips and mouth. By 30 minutes, there is extensive muscle weakness, plummeting blood pressure, and severe paralysis. Death can occur within this short half-hour window. If they happen to survive longer, respiratory failure gradually sets in over 6 hours. Did you know a victim's cognition often remains entirely clear while their physical body completely paralyzes around them? It's horrifying. There is no known antidote. Clinical management relies purely on symptomatic respiratory support.
This exact mechanism brings us to a fascinating application in forensic science. Chemical analysis of powders used in traditional Haitian rituals to induce a so-called zombie state revealed exceptionally high levels of this exact compound, Tetrodotoxin. By carefully controlling the dose, practitioners could induce a profound, semi-conscious, paralyzed state that closely mimics actual death. It is a stunning, real-world example of ethnopharmacology intersecting directly with modern analytical toxicology.
Section 4. Toxic tides and shellfish. Let's transition to another deadly marine phenomenon that regularly disrupts commercial food supplies globally: toxic algal blooms and their dangerous effect on shellfish. Take a look at this dense cluster of mussels. Normally, these are innocent, filter-feeding mollusks. They naturally process large volumes of water to feed. However, during warmer weather, certain microscopic, single-cell dinoflagellates proliferate rapidly. This causes a red-brown discoloration in the water, commonly known as a red tide. When our favorite mussels, oysters, and clams filter these toxic algae, they suddenly become highly poisonous to humans. This triggers what we call paralytic shellfish poisoning, or PSP.
The primary toxin responsible for PSP is Saxitoxin. Now let's compare Saxitoxin to the Tetrodotoxin we just discussed. Both are immensely potent. Both cause severe paralysis by blocking voltage-gated sodium channels. However, analytical chemists discovered something truly remarkable here. A pufferfish is naturally immune to its own Tetrodotoxin, but if that same pufferfish is exposed to a red tide, it is completely vulnerable to Saxitoxin. Why is that? Because Saxitoxin blocks the sodium channel at a completely different binding site. It's a brilliant nuance of receptor pharmacology.
Section 5. Ciguatera, the reef mystery.
Let's move a bit further up the marine food chain to explore a highly unpredictable syndrome found in large tropical reef predators. This is a massive hurdle for food safety in regions like the Caribbean, Hawaii, and the South Pacific. The toxin, Ciguatoxin, is produced by a dinoflagellate called Gambierdiscus toxicus. Herbivorous fish eat the algae, and then larger carnivorous fish, like your groupers, snappers, and barracudas, eat them. The toxin bioaccumulates. It builds up. It causes severe gastrointestinal and neurological symptoms. A defining clinical marker for diagnosis is something called cold allogenia. This is where simply touching something cold actually triggers a severe burning sensation. Now, for the food technologist tuning in, here is the real headache. Ciguatoxin is completely odorless, it's tasteless, and it is highly stable. You simply cannot cook it out. It easily survives the frying pan intact.
And this brings us to a fascinating, unifying revelation about our explainer today. As leading researchers have noted, Tetrodotoxin is actually not made by the fish. It's made by relatively common marine bacteria that associate with these animals. The pufferfish, the shellfish, they merely absorb it from their environment. The animals themselves are just the vessels. The microscopic ecosystem is the true manufacturer.
Which leaves us with a provocative, vital question for all scientific disciplines moving forward. We know that toxic dinoflagellates and marine bacteria thrive in warmer waters. As climate change steadily warms our oceans and expands these microscopic bacterial blooms, what new hidden toxins might infiltrate our commercial food supply next? It is a profound, pressing challenge for toxicologists, analytical chemists, and food scientists worldwide. Thank you so much for joining me in analyzing this fascinating science.